Greetings everyone, it's good to be back again with another video for Form 4 Biology. In the last video, we studied the movement of substances across the plasma membrane, part 1, where we studied simple diffusion and osmosis. So this video is a continuation of the different types of movement of substances across the plasma membrane, and we'll finish up with these two, facilitated diffusion and active transport. Here is a schematic diagram that we used in the last video to talk about the movement of substances across the plasma membrane. So, movement of substances across plasma membrane can be divided into two types. Passive transport, where molecules move down the concentration gradient, and this does not require energy. Then active transport, where molecules move against the concentration gradient, and this needs energy. Okay, where the cell has to actively produce the energy. That's why it's called active transport. Now, passive transport does not need energy and is divided into three types. Simple diffusion. Osmosis is just the diffusion of water across a semi-permeable or selectively permeable membrane. And then we have facilitated diffusion, which uses either carrier proteins or channel proteins to help the molecule be transported across the plasma membrane. So for simple diffusion and osmosis, molecules move through the phospholipid bilayer, such as water for osmosis, and then for simple diffusion, vitamins A, D, E, and K, which is lipid soluble, can pass through the phospholipid bilayer, and also lipid soluble molecules like fatty acids and glycerol. For facilitated diffusion, molecules that use these two, the carrier proteins, are like glucose, and ions are used. Ions use the channel proteins to move past the plasma membrane. For active transport, there's only one type. It uses carrier proteins and it needs energy. So for this video, we'll just study facilitated diffusion first, because if I were to make a video with these two, it will be too long for facilitated diffusion and active transport. So this video is more about facilitated diffusion, and in the last video, part 3, we will discuss active transport. What is the function of facilitated diffusion? Well, there are certain molecules that cannot pass through the hydrophobic region of the phospholipid bilayer. This hydrophobic region contains the hydrophobic tails of the phospholipid molecules, and it is non-polar. It repels water too, so it will attract those of like nature with it, and those that are different from it, it will repel or will not allow to get near. So that's why for hydrophobic region, they allow the lipid soluble substances to pass through them. It will dissolve in the hydrophobic tails first, and then the lipid soluble substances will pass through. But what about the lipid insoluble molecules, such as the charged ions, large polar molecules such as amino acids and glucose? They are all unable to pass through the phospholipid bilayer. And if it's a polar molecule, it has uneven distribution of charges, so there'll be one end that's negative and one end that's positive. All these substances cannot get through the phospholipid bilayer, so the other alternative is to use the transport proteins, like this channel protein and this carrier protein that can change shape to move across the plasma membrane. The process involved is called facilitated diffusion. In other words, it is a form of assisted diffusion using the channel protein and the carrier protein. Let's look at some characteristics of the transport proteins. Firstly, the channel protein. It has a channel or pore or canal to allow the charged ions and small size solutes to diffuse through across the plasma membrane. So small size solutes and charged ions, they are small, because the pore is quite small, so it can only allow the small ions and small molecules to pass through it. Furthermore, each channel, like this one here, has specific internal characteristics that allow only specific ions to pass through it. So it's specific in its function. Maybe this pore protein is used to transport calcium ions. So it is specifically for the calcium ions to move through the pore. So it's specific in its function. For the carrier protein, it has a special binding site that will bind to a specific molecule only. So maybe we have carrier protein specifically for amino acid molecules. So it will only bind to the amino acid molecules. And the site, the amino acid molecules can only bind to the specific site on the amino acid carrier protein. So even the site is specific. So a carrier protein that transports the amino acid molecules across the plasma membrane cannot transport other molecules like glucose across the plasma membrane. So it's specific in its function. The first type of facilitated diffusion uses the channel protein. Let's discuss this as a question. Let's say you're given this question in the exam. Based on the diagram above, explain how the molecules move into the cell. So you got to analyze the diagram first, and we see that this is something like a channel protein with the channel in the center. Next, we can see that there are many molecules outside the cell, and inside the same type of molecules are less. So these molecules are moving down the concentration gradient from a region of high concentration to a region of low concentration. So this is a type of passive transport, and it is actually facilitated diffusion because it uses a channel protein. So to answer the question, first we mention the type of movement. The process involved is facilitated diffusion. Next, we have to explain the whole process. So always start out with the concentration. Concentration of the substance is more or higher at one side than the other. So concentration of, now here is labeled chloride ions. So we use chloride ions. Could be calcium ions and other ions, charged ions. The concentration of the chloride ions is higher in the extracellular fluid than in the cytoplasm of the cell. Okay, so talk about the concentration is higher somewhere than in another place. Then mention how the chloride ions diffuse. So they diffuse and move through the channel of the channel protein into the cell. And the third point, you can mention ions move down the concentration gradient. And fourthly, no energy is required for this process. So you have 4 or 5 points here to get your marks. Always write a little bit more, extra 1 or 2 points, in case some points are wrong. The next type of facilitated diffusion is the one using carrier protein. So let's discuss this. Here we have the diagram of the carrier protein, and the glucose molecule is bound to it here. This is the inside of the cell. Okay, so let's say you have the same type of question, like based on this diagram, discuss how molecules are transported into the cell. This molecule, what molecule? Okay, so again, you can mention first that this process is facilitated diffusion using the carrier protein. Okay, then next, talk about the concentration. Concentration of glucose molecules is higher in the extracellular fluid out here than in the cytoplasm of the cell. B, what happens to the glucose molecule? So it will bind to a specific binding site on the carrier protein here, Okay, and the third step, the carrier protein will change shape. So this is to allow the glucose molecule to pass through, like this, and like this, right? So it passes from outside the cell into the cell. So the carrier protein changes shape to allow the glucose molecule to pass through, very important, and enter the cell. And after that, it will return to its original shape to transport more glucose molecules into the cell from the extracellular fluid. So the main thing is talk about the concentration of the glucose molecules, higher in which part, and talk about the change of shape. The carrier protein can change shape. And thirdly, talk about that it returns back to its original shape. And also, fourthly, no energy is required for this process. We have already discussed facilitated diffusion using channel protein and carrier protein. Now let's discuss the last type of movement of substances across the plasma membrane, and that is active transport, which is very different from the other three types of movement. It's very different from simple diffusion, osmosis, and facilitated diffusion in that active transport requires the use of energy. So let's look at the definition of active transport, which we have to memorize. Active transport is the movement of molecules or ions from a region or area of lower concentration to a region or area of higher concentration. So from a region of lower concentration to one of higher concentration, and this is something like the analogy of a man climbing up a hill from a lower place to a higher place. This needs the use of energy. The man has to use energy to climb up the hill, as compared to when going downhill, he needs less energy. So for active transport, moving the molecules or ions from an area of lower concentration to an area of higher concentration is not a natural process. It's going against the concentration gradient, and therefore it needs the use of energy. And it must occur across a plasma membrane which has carrier proteins. It only occurs in living things. So because it requires this energy that has to be released from the breakdown of a molecule called adenosine triphosphate, which we'll study later. So ATP is adenosine triphosphate, and it is a molecule that provides immediate energy or instant energy when it breaks down to form ADP and phosphate group. So this energy is then used for active transport and for muscle contraction and other activities of the cell. Now this ATP is produced during cellular respiration in the mitochondria. As we know, cellular respiration is the process whereby glucose is oxidized to release energy and also to produce water and carbon dioxide. So the energy released from cellular respiration is used to form this ATP, which then breaks down when the cell needs to use energy for active transport. Other than ATP, the cell also needs to use specific carrier proteins which have to transport the substances against the concentration gradient. So notice that all the terms here begin with A, active transport, against the concentration gradient, across the plasma membrane, use of ATP. 4 A's for active transport that you have to remember when you explain active transport, and a C for carrier protein. Let's have a look at a carrier protein used in active transport. So this carrier protein has 3 binding sites for 3 sodium ions to bind to it, and these sites are specifically for sodium ions only to bind to it. Okay, so this carrier protein will transport the sodium ions out of the cell, and this part here is to bind to another ion, which is potassium ion. Okay, then ATP is needed So when ATP breaks down to form ADP and phosphate group, the phosphate group will then provide the energy to the carrier protein to change shape and transport the ions out. So this carrier protein will open up to the extracellular fluid to release the sodium ions to the extracellular fluid. What actually is ATP? ATP stands for Adenosine Triphosphate, A, T, and P. Let's look at the structure. ATP consists of adenosine, this molecule here, and 3 phosphate groups. It is the last bond here between two phosphate groups that is energy rich. If this bond is broken, especially when ATP is broken down and this bond is broken, then the energy is released for the cell to carry out its activities. So we can say that ATP is a molecule that stores energy in the cells. And when the cell needs the energy for activities, it will break down immediately to provide an immediate source of energy for active transport, muscle contraction, nerve impulse transmission, and so forth. So it provides energy when the phosphate bond is broken. How is it formed? So it is formed using the energy generated from cellular respiration when glucose is broken down to produce energy, water, and carbon dioxide. This occurs in the mitochondria, cellular respiration, and then that energy from the breakdown of glucose is used to synthesize the ATP molecule in large amounts for the use of the cell. These 3 equations show us the formation of ATP and the breakdown of ATP. Starting with cellular respiration, where glucose is oxidized by the cells using oxygen and is broken down into carbon dioxide, water, and energy. So this energy that's released from the oxidation of glucose is not used for muscle contraction or active transport directly. It must be stored in the ATP molecule. To form the ATP in the cell, there will be adenosine diphosphate, ADP, and there will be phosphate groups. So ADP combines with phosphate and forms a chemical bond with it using the energy provided by cellular respiration, and we get ATP, the molecule that stores energy in the chemical bonds between the 2 phosphate groups. Okay, so there are lots of this molecule in the cell. For example, in muscle cells, there are a lot of ATP molecules waiting to be broken down to release the energy for activities like muscle contraction. So this ATP, when it breaks down, will form again the ADP, adenosine diphosphate, and phosphate groups. And the energy is released from the breakdown of the phosphate bonds. Then this energy is directly used for active transport, for example, to change the shape of the carrier protein for active transport and for muscle contraction and nerve impulse transmission. So we say that ATP is the currency of the cell. It uses it to get energy and then it will use it to perform its activities. This diagram shows you the cycle where ATP is formed and then broken down to form ADP and then formed back again. So this process occurs in the cells all the time. Now ATP can be broken down quickly to release energy for work and form back again by using energy from oxidation of food, that is from cellular respiration. So we have ATP here, adenosine plus 3 phosphate groups. This last bond is the one that's energy rich. So if it is broken down, it will release the energy for cellular activities, including active transport. And 1 free phosphate group is also produced. Then the cell will form back the ATP using energy from cellular respiration. So ADP, which is adenosine plus 2 phosphate groups, will form a bond with the free phosphate group using the energy from cellular respiration, and then this produces ATP once again, which can then be used over again to provide the energy immediately for cellular activities. Let's discuss the carrier protein that's used for active transport. So carrier protein used in active transport of substances is a specific carrier protein with specific sites to bind with specific molecules. It's very specific. And on the carrier proteins, there are receptors also to bind with the ATP molecules. As you can see here, this carrier protein is to transport sodium ions out of the cell. So these are the specific sites that are used to bind to the sodium ions. No other ions can bind with the carrier protein at these specific sites. They're only for the sodium ions to bind to it. Whereas on the other side are the specific binding sites for potassium ions, only for potassium ions. Then on the carrier protein, we can see receptors here that allow the ATP to bind to it, the phosphate group especially, after ATP has broken down or split. The phosphate group will bind to the carrier protein at this receptor here. Next, when a phosphate group attaches to a carrier protein, it will provide energy for the carrier protein to change shape. So here we see ATP already broken down or split into ADP and phosphate. The phosphate group will attach itself to the receptor on the carrier protein, and the energy that was released from the breakdown of ATP will be given to the carrier protein, and it will change shape so that instead of facing downwards, it will open up to the outside, to the extracellular fluid, and release the sodium ions to the extracellular fluid. So in this way, the molecules or ions will move across the plasma membrane, in this case out of the cell. Let's talk about the term carrier protein and how they are associated with pumps. Active transport actually results in either the accumulation of molecules in the cell or the excretion or elimination of molecules or ions from the cell. So in active transport of a substance, there is no dynamic equilibrium achieved where both the region inside the cell and outside the cell has the same concentration of that molecule X, for example. Meaning to say that in active transport, the cell is either accumulating the ions in its cytoplasm, so it will have more of these ions or molecules in its cytoplasm, much more than in the extracellular fluid, or it may be excreting the molecules or ions from the cell. That means the cell will have very low concentration of these molecules or ions compared to the external environment. So it's either high concentration of the substance in the cell compared to the outside, a higher concentration inside and outside, or a much lower concentration of the substance in the cell compared to the outside. Okay? Thus, we can say that carrier proteins involved in active transport are like the pumps, like water pumps. For example, if a house has been flooded during the floods, sometimes the water is hard for the water to flow out because maybe the house is lower than the ground outside. So water pump can be used to pump the water out against gravity, pump it out and then let it flow into a drain. So likewise, carrier proteins, they act like pumps in that they pump the ions or molecules either into the cell or out of the cell against the concentration gradient. Example of pump is a pump is in the animal cells, and this is called the sodium potassium pump in the nerve and muscle cells. The sodium potassium pump is actually a carrier protein that transports sodium ions and potassium ions against their concentration gradients across the plasma membrane. Okay, as you can see in this picture here, this carrier protein is the sodium potassium pump. On one side, it binds with sodium ions and transports them out of the cell. It pumps the sodium ions out of the cell against the concentration gradient, and on this side, it binds with potassium ions and brings them into the cell. So it accumulates the potassium ions, but it eliminates the sodium ions. This means that sodium ions are always transported out of the cell into the extracellular fluid, so the concentration of sodium ions in the cell is much lower than in the extracellular fluid. On the other hand, potassium ions are always transported from the fluid, extracellular fluid, into the cell. So the concentration of potassium ions is much higher in the cell than outside the cell. So here's a song that you can sing to relate to the function of the carrier protein as a pump. Okay, and the carrier protein needs to use energy from ATP to change shape and pump the ions either into the cell or out of the cell. Okay, so this song is following the tune of London Bridge is Falling Down, and I composed this song just to help you remember some of the characteristics of the pump, in this case the sodium potassium pump. So let's sing it together, and it's following the London Bridge song. Okay, it goes like this. Hey ATP, please come, help me be a good pump. Potassium in and sodium out, change shape in and out. Simple, isn't it? Okay, it's a biology song. And here we have ATP responding by saying, okay, carry is the name of the carrier protein. Splitting means it's breaking down to form the ADP and the phosphate group, which provides the energy for the carrier protein to change shape. Let's discuss the mechanism of the sodium potassium pump, which is a carrier protein that carries out active transport. So these explanations here are also for the active transport of sodium ions. In the first stage, 3 sodium ions bind to the binding site of the carrier protein, which has 3 binding sites for 3 sodium ions to bind to it. Next, ATP in the cell breaks down to form ADP and P, the phosphate group. The phosphate group then binds to the carrier protein in order to provide energy to it. In the third stage, the phosphate group has provided the energy for the carrier protein to change shape. So it will change shape and face the external environment or the extracellular fluid. In this way, the sodium ions can be released from here and they are transported out of the cell. So this is not the end of the transport of ions, because now the cell has to take in the potassium ions on this side and transport it into the cell. So to continue with the process of active transport, the same carrier protein that was used to transport the sodium ions out of the cell is used to transport the potassium ions into the cell, again by active transport. So, but then this site now is used. The binding site is different. The binding sites for potassium ions are on the opposite side here. The first point is that the potassium ions from the extracellular fluid outside will bind to the carrier protein. And notice that the concentration of potassium ions is lower outside the cell than inside the cell, yet the cell wants to take in more potassium ions, okay, from a region of low concentration to a region of higher concentration in the cell. So potassium ions from extracellular fluid bind to the carrier protein, and then the phosphate group. Okay, please don't confuse the phosphate group with potassium. Okay, potassium ion is the one that needs to be transported. The phosphate group was the one that provided the energy for the carrier protein to change shape just now. But now it doesn't have to provide any more energy. It just leaves the carrier protein. And with the loss of the phosphate group, the original shape of the protein, carrier protein, is restored. So it will face downwards now. Okay, from facing upwards, it's facing into the cell. So when it changes shape here, it goes back to its original shape due to the loss of the phosphate group. It will be able to allow these potassium ions to be transported and released into the cell. Okay, so for this process of active transport of potassium ions, the energy was not needed here, okay, as in the first part for the transport of sodium ions. Let us discuss another pump or carrier protein that is called the proton pump. So the word proton here means the hydrogen ion. So this pump is a carrier protein found on the epithelial cells lining the stomach cavity here. So inside the stomach, in the cavity, and on the surface of the wall here, we have the epithelial cells. So the carrier protein is found on the plasma membrane of the epithelial cells, and it's called the proton pump. Its function is to cause the stomach contents to become acidic with the production of hydrochloric acid. What is the mechanism whereby the hydrogen ions are eliminated from the cell so that it can form the acid in the cavity here? So firstly, the hydrogen ion inside the cells of the epithelial tissue will bind to the site of a carrier protein of the cell. Okay, so this is the proton pump or the carrier protein. The hydrogen ion will bind to the binding site of this proton pump, and next ATP will break down to release the energy in order for the carrier protein to change shape. So as it changes shape, you will be able to transport the hydrogen ion out of the cell. It's not shown here. These are the hydrogen ions against the concentration gradient from region of low concentration to region of high concentration. In this way, the hydrogen ions will accumulate in the stomach cavity, and they will form the hydrochloric acid, which destroys bacteria and other pathogens in the food that we eat. So that is the function of the proton pump, and also the mechanism to produce the acid. By pumping out the hydrogen ions. That's all for this video. In the next lesson, we'll continue to study Chapter 3, 3.3 and 3.4 about the movement of substances across the plasma membrane in living organisms and its application in daily life. Thanks for watching. Stay tuned for the next video, and share, like and subscribe.